Okay, so now we're going to be talking about human blood groups. And when we're looking at human blood groups, we're just going to be looking at the ABO allele, or gene, I'm sorry, which is going to just have three alleles, the A allele, the B allele, and the O allele. So this is a system that is codominant. So type A and type B are codominant. O is going to be the recessive. But now instead of having two alleles, we have three alleles, and we have codominance in this system, so it makes it a little bit more complicated. When we're looking at what these mean, when it says you have type A blood, it means that on your little blood cells here, you've got A proteins, or proteins that are kind of nicknamed A's. So this is type A blood. Type A blood. So type B blood, then, is going to have B proteins hanging out on those blood cells. AB blood, so somebody who's type AB, they're going to have both A proteins and B proteins on their red blood cells. Type O, again, remember type O is the recessive, they're not going to have any proteins. Type O is the lack of proteins on those red blood cells. So that's what these look like. Now, if you'll refer to the chart under human blood groups in your lab manual, you'll see the blood type, the genotype, and the phenotypes. So what we're seeing here, these are the phenotypes. So type B blood has the B proteins, type A blood has the A proteins, type AB blood has both, type O has none. So type A blood could be genotype AA, two A alleles, or AO, one A and one O, again with O being the recessive. B, same thing, BB or BO. Type AB blood only has the one option, the codominant, AB. And type O also only has the one option, type OO, okay, two O alleles. In terms of inheritance, it works the exact same way with our Punnett squares. So we could do a Punnett square, let's say we're crossing a type A individual, so someone who's heterozygous A, so AO, with someone who's, let's say, type B, and let's say homozygous B, so BB. Okay, so we're going to cross those two individuals, so we put our AO at the top, we put our BB on the side, again, AB, bringing down and coming across, AB, then we've got BO and BO. Okay, so these two parents would only be able to produce type B offspring or type AB offspring. Right? And we could do that no matter which set of parents we're looking at. We can also kind of work our way out. So if we know that an offspring, let's say, is type AB, this is the child, then we know one parent, okay, so parent 1 and parent 2, because we don't really know which one's the mom and which one's the dad, we know one of them has to have an A allele and one of them has to have a B allele. Okay, but we don't necessarily know what that other allele is. So this parent could be AA, could be AO, could be AB. Same thing with this parent. This parent could be BA, which would be our AB, BO, or BB. We don't know what their other letter is to have this child. But let's say this same couple has a second child that's type BB. Okay, then we know that this parent has to have a second B allele. So couldn't be AA, couldn't be AO, has to be AB, whereas this parent still could be any of the three because we just need the single allele. So we could build that in a Punnett square working our way out. We would put our AB in one spot. That's where we got our A and our B originally. And then we would put, let's say, our BB here, using that same B allele, but we would know that that would have to be there. Now, if we had, let's say, a third child that's BO, okay, maybe in this case, we'd have to have the O here to have our BO child here, meaning this parent is type BO, no A, no B. Okay, so we can work from the inside out or the outside in when it comes to Punnett squares, which actually will lead us into our concept of pedigrees in our next chapter. We can determine the type of blood that somebody has based on how the blood reacts with antibodies, which will be the next topic that we cover.